So for our new Grans season, we have really a fantastic opening lecture today, and that's Dr. Rhian Touyz. And she's the Executive Director and Chief Scientific Officer of the Research Institute at McGill University Health Center. She's also Dr. Phil Goh Chair of Medicine at McGill University. She was recruited to Montreal in 2021 after serving 10 years as the director of the Institute of Cardiovascular and Medical Sciences, a British Heart Foundation chair and professor of cardiovascular medicine at the University of Glasgow in Scotland, the UK. And we had a chance, to do a lot of collaborative work. It's just a fabulous center. And from selfish point of view, Rhian could not have come back to Canada at a better time. We were previously wanted Rhian, to really help us on our large grant application, bring heart interconnectome as an international collaborating center from University of Glasgow. So imagine our delight when we found out Rhian was actually coming back to Canada to McGill Research Institute as a director. So we quickly, was able to have Rhian as a partner with us, including the institution of McGill as a full partner with our application. Of course, our success, a lot of can be credited to Rhian's inspiration, wisdom, and numerous feedback on our application. Dr. Touyz is a clinician scientist, received her MD, PhD from South Africa, and was recruited to Canada as a postdoctoral fellow at the famed Clinical Research Institute of Montreal, ICRM. And she was actually on staff before being recruited to the Kidney Research Center here, Ottawa Hospital Research Institute, and where she was Tier 1 Canada Research Chair in Hypertension. And I was introduced to Rhian many years ago through Dr. Ernesto Schifrin, who was on my Institute Advisory Board when I was CHR. Rhian was already a star researcher under unraveling the mechanisms of hypertension, but also work on the SHIP Guideline Committee, who I work together with Sheldon Tope subsequently, but Rhian was the co-chair, to lead that effort. And that really puts hypertension control in Canada on the world map. She also played a major leadership role in premier hypertension and cardiovascular organizations. She was president of Canadian Hypertension Society, chair of the High Blood Pressure Research Council, American Heart Association, president of International Society of Hypertension, president of European Council of Cardiovascular Research. And she was also, importantly, the founder chair of the Women in Hypertension Research Network of the International Society of Hypertension. And this is a very important mission which encourage, support, inspire women to take a career in science and medicine and research. And she has mentored over 100 trainees, many of whom are really successful leaders today. Dr. Touyz is current editor-in-chief of Hypertension and past editor-in-chief of Clinical Science. She's advisor to Max Delbrück Center in Berlin, INSERM in France, Mayo Clinic, Cardiovascular Research Center in the US, and MRC and Wellcome Trust in the UK. Had a chance also to really admire the impressive growth of the Institute of Cardiovascular and Medical Sciences in Glasgow, led by Rhian as part of the BHF Center of Excellence. And I was serving as the International Advisory Panel, and it was really very impressive to all the panel members of her accomplishments. Despite her multiple roles, she lead exemplary research on the molecular mechanism of hypertension, small vessel disease, was particularly interested in vascular signaling and the redox biology of cations and trip channels, and also cardiometabolic disease, including obesity and adipose dynamics. And her work really in small vessel disease is pioneering. I think now it is really coming into the forefront. And her research is translational, including proteomics, cellular molecular biology, transgenic models, clinical imaging, clinical studies, including the important work on human physiology that actually validates a lot of these mechanisms. She's published over 575 peer-reviewed papers and received multiple awards. And for her exceptional accomplishment, she has being awarded the Dow Award, as well as Harriet Destin Research Award from American Heart Association, Distinguished Scientist Award from Hypertension Canada, Irving Page Award, American Society of Hypertension, Joan Mott Award from Physiological Society, and Research Excellence Award and Premier International Award from Council of Hypertension, or AHA. So Rhian is truly a leader and pioneer, and also someone who actually inspires us all in research. And we're absolutely delighted and privileged to have Rhian today to deliver the opening lecture for our Grans season, and is entitled "The Vasculome in Hypertension and Small Vessel Disease." Rhian.
Peter, thank you so much for that really very, very kind introduction. And it's just so lovely to be back. I remember standing here maybe 15 years ago when I was still working at the Kidney Research Center. We used to come and have meetings here. So it's really lovely to be back. Thank you for the invitation. And let me see. Okay, so indeed, I have spent the last 25 years of my career studying hypertension. Why? Why have I been studying hypertension? Well, it might be surprising for you to note that the global burden of disease, whether this is related to premature death or to morbidity, the major risk factor underlying this worldwide is high blood pressure. It's really amazing to understand this, because many believe that cancer, trauma, infectious diseases are the major cause of, or the major risk factors for the global burden of disease, but it actually relates to high blood pressure. And because of this, there's been huge efforts, obviously, in trying to better control hypertension in populations. And as we can see here, data from the National Haynes study, over the past 10 and 15 years or so, from the 1990s, we were doing pretty well in terms of improving blood pressure control. But more recently, as we can see, since about 10 years ago, the trajectory of blood pressure control, both in the United States and in Canada, as well as in Europe, is actually declining. So this poses a huge problem in terms of the impact down the line in terms of the morbidities associated with hypertension. And actually, it was even in the 2000s that Avram Chabanian, in his Shattuck lecture, mentioned to us that the treatment of hypertension should be one of medicine's major successes, because the advances in therapy provide capacity to lower blood pressure in almost every patient. However, we have uncontrolled disease despite improved therapy, and he termed this the hypertension paradox. And the reason why the impact of hypertension is so great globally, as we know, is that high blood pressure is the major risk factor for many of the chronic diseases that burden our society today, including cardiovascular disease, vascular dementia, kidney disease, etc. So this is what Chabanian said in 2009. He called it the hypertension paradox. Where do we stand today in 2023? Well, not much has changed. If anything, things are getting worse, as I just showed you, with respect to the control rates of blood pressure in many of the countries that are tracking blood pressure control. So why is this? Well, indeed, it's true. We do have very effective blood pressure medication, antihypertensive drugs, but the pathoetiology of hypertension is actually very complex. And it's really hard to imagine that in 95% of patients, we really don't know the exact cause of hypertension. Nevertheless, it was in the 1960s that Rhian Touyz put together his mosaic, where he began to identify a number of factors that actually interacted with each other that were responsible for blood pressure regulation and the pathogenesis of hypertension. So that's what the mosaic looked like in the 1960s. However, more recently, we know that this is much more complex. If we start adding to this mosaic the complexity of the genomic factors that have been implicated in hypertension, we've had to go back to that mosaic and re-look at it and actually include in that original mosaic of PAGE a number of new factors that are emerging as being very important in the underlying factors associated with the development of hypertension. In particular, I would like to stress to you that the vasculature is critically involved, the activation of the renin-angiotensin-aldosterone system, and increasingly we're beginning to appreciate the importance of oxidative stress and inflammation. So let's go back to our very basic principles of the physiology of blood pressure control. We know that the kidney is very important, and the very famous sodium natriuresis curves, which are abnormal in patients with hypertension, play an important role in controlling sodium volume homeostasis. But we also know that mean arterial pressure is the product of cardiac output and total peripheral resistance. And at least within the context of essential hypertension, it is an increase in total peripheral resistance that is primarily responsible for an increase in blood pressure. And the factors most important for an increase in total peripheral resistance are those small microvessels, the peripheral resistance vessels, that play a role in contributing to an increase in total peripheral resistance, either through increased constriction, reduced dilation, or vascular remodeling. So my career has really been spent in trying to understand the changes that occur in those small resistance arteries in the context of hypertension. And our research, together with many, many other contributions, have now identified what we term the vascular phenotype of hypertension. We see a normal vessel, as we can see, a clean vessel, normal endothelium, a small vascular media. What happens in hypertension is we get endothelial dysfunction and inflammation. We get an increase in the size of the vascular media. The vascular smooth muscle cells become disorientated, and we start getting inflammation and fibrosis. And this is what we term the phenotype of hypertension. Now, many years ago, work that we did actually together with Ernesto Schöpfen at the IRCM is that we found that this process is actually reversible if blood pressure is properly controlled with certain antihypertensive drugs. But we were very interested to know what are the underlying molecular and cellular processes that contribute to this vascular phenotype in hypertension. And again, we and others start to dissect out the many processes that underlie this phenomenon. But something that was very fundamental to these processes in hypertension was activation of the renin-angiotensin-aldosterone system. And so we started to look at the signaling pathways that underlie these processes, especially linked to activation of the renin-angiotensin-aldosterone system. And when we started to put together the signaling molecules that are activated or dysregulated in hypertension, link them then to the renin-angiotensin system, one of the factors that came out as being especially important in terms of this network was actually reactive oxygen species. And about 15 years ago, the significance of reactive oxygen species and oxidative stress started to become more evident in the context of cardiovascular disease and in hypertension. And this was just one study in human hypertension that showed us that as blood pressure increased, so too was there an increase in mediators or indices of reactive oxygen species in patients with hypertension. So for those of you who may not be familiar with what reactive oxygen species are, from a very simplistic point of view, reactive oxygen species are any intermediates in a redox process leading from oxygen to water. And within the context of cardiovascular disease and the cardiovascular system, those reactive oxygen species that are most important are superoxide anion, hydrogen peroxide, as well as the important vasodilator, nitric oxide. And indeed, when we started to probe the generation or the presence of reactive oxygen species in small vessels from patients and experimental models of hypertension, not only did we see that these vessels underwent marked vascular remodeling, as we can see here, by this very thickened wall in these rodents that were induced by angiotensin, but we can see associated with this was an increase in the bioavailability of reactive oxygen species compared to normotensive models. And this indicates oxidative stress. This is a fluorescence marker that is actually detecting reactive oxygen species in the vascular wall. And there have been many studies to show that vascular generation of reactive oxygen species is increased and dysregulated in hypertension. So of course, the real question is, what is it that is responsible for this increased generation of reactive oxygen species? And again, about 15 years ago, the NADPH oxidase system in the cardiovascular system became very evidence of importance. And the NOx world started to increase, or there was an increase in research related to the NOxes and the vascular system. And there are 7 NADPH oxidase isoforms. NOx2 is the prototype, which is typically found in phagocytes, but there has been evidence to show that NOx1, NOx2, and NOx4 do play some role in the inflammatory processes and remodeling in experimental hypertension. But of the 7 NADPH oxidases that have been identified, it is NOx5, which is the newest of this family of NOxes that has been identified, that is especially important in the context of human disease. Now, as you can see, these NADPH oxidases are very complex oxidases. They need a whole lot of subunits in order for them to be activated. But NOx5 is very special in that it is quite dissimilar in its structure and its dependency on other NADPH oxidases for its activation. And we termed NOx5 the enigmatic NOx. And the reason for that is that NOx5 is very sensitive to intracellular calcium. So an increase in intracellular calcium results in an increase in activation of NOx5 and the production of superoxide. NOx5 is activated through conformational changes. It doesn't require the NADPH oxidase subunits. It is the first of the NOxes to be crystallized. And most importantly for us, where we rely a lot on experimental models in the lab, NOx5 gene is absent in most rodents, including rats and mice. And so it becomes very challenging to study NOx5 in the laboratory. But what we started to ask is if NOx5 is so important with respect to its activation by calcium, and we know that calcium is intricately involved in normal vascular function, cardiac function, may there be an association between calcium, NOx5, and reactive oxygen species in the context of hypertension and vascular dysfunction, as I've explained to you, defining the phenotype. So many years ago, when I was still here in Ottawa working in the Kidney Research Center, we started to characterize NOx5 in various pathologies, starting with diabetic nephropathy, diabetic vasculopathy, atherosclerosis. And when we started to probe the presence of NOx5 in tissues from patients, indeed, we found that NOx5 is highly expressed in pathological conditions and is associated with an increased production of reactive oxygen species. We also showed that changes in calcium activate NOx5 and play a critical role in the production of reactive oxygen species in the vascular wall. So we have gone on now to really identify through high fidelity phenotyping the role of NOx5 and reactive oxygen species in the pathophysiology of vascular biology in hypertension. We have used a number of models, and because our lab focuses a lot on translational research, we rely as much as possible on studying the vasculature from very carefully phenotyped patients, both normal, healthy individuals and patients with hypertension. We isolate small vessels through a gluteal biopsy technique. We look at the vascular function of these small vessels, and we also culture from these vessels vascular smooth muscle cells, fibroblasts, adipocytes, where we can then go on to do some of our molecular and cellular studies. And I'll get onto this in just a minute, but we've also gone on to generate some very interesting mice. So what did we find? Indeed, we found that if we looked at these small vessels from patients with hypertension or normotensive individuals, there is a really quite heavy expression of NOX5 in the vascular media. When we isolate the vascular smooth muscle cells, expose them to angiotensin II, and look at the production of reactive oxygen species, NOX5 plays a very important role in the production of reactive oxygen species, because if we inhibit NOX5 with a NOX5 inhibitor called melatonin, the production of reactive oxygen species is reduced. But more intriguingly, when we looked at where NOX5 is localized in vascular smooth muscle cells, it was very clear that it has a very specific localization in intracellular compartments. NOX5 is found in the plasma membrane, as we can see, but especially in hypertension, in vascular smooth muscle cells from hypertensive individuals, we see a marked expression of NOX5 in the endoplasmic reticulum. And actually, if we consider that the endoplasmic reticulum is a site where it's very rich in calcium, and it's also a site when proteins undergo post-translational modification, it does make some sense that NOX5 may be regulated there. And so we then started to ask the question, what is the functional relationship between the ER, NOX5, and oxidative stress? And similar to what others had found, when we started to probe these vascular cells from normal and hypertensive individuals, looking at markers or indices of ER stress, there was a huge signal for ER stress in the vascular smooth muscle cells from hypertensive individuals. And moreover, when we induced ER stress with tunicamycin, the expression of NOX5 was increased. And so it really suggested to us that the ER is somehow playing some role in the regulation of NOX5 and its production of reactive oxygen species. So this is just a cartoon to show you some of the work we've done, that NOX5 is in the ER, it's activated probably through calcium, reactive oxygen species are produced, and we know that reactive oxygen species themselves play a role in the ER stress response, the unfolded protein response. So there is a biphasic interaction between NOX5, oxidative stress, and the ER. But we also found that NOX5 is localized in the plasma membrane. And it's in the plasma membrane that NOX5 actually associates with signaling molecules that are responsible for cytoplasmic redox signaling. The question is, how does NOX5 traffic from the endoplasmic reticulum to the plasma membrane? And very importantly, the plasma membrane, where it associates with cholesterol-rich microdomains, really targeting to lipid-rich foci. So working with colleagues in Glasgow when I was still there, we started to look at the structure of NOX5, and what became evident is that in its post-translational modification of NOX5, it contains a number of cysteines which are very tightly regulated by palmitylation. Now, as you know, palmitylation is a lipid form of post-translational modification, and we thought that this was particularly interesting because it might play a role in associating with some of the lipid-rich microdomains. So working with Professor Will Fuller, who's an expert on palmitylation, he generated cells that were deficient in palmitylation, and we started to look at what happened to the production of reactive oxygen species in the context of NOX5 activation. And what we can see is that in normal cells which have normal palmitylation, and when they're stimulated, they get an increased production of reactive oxygen species, an effect that is markedly attenuated when these cells are deficient in palmitylation. So NOX5 palmitylation seems to be very important in the regulation of this oxidase and the production of reactive oxygen species. Similarly, Will showed that the palmitoylated form of NOX5 very closely associates with the endoplasmic reticulum, something that is deficient when you have the mutant form of the palmitylation. And moreover, Will went on to show that the NOX5 membrane trafficking from the endoplasmic reticulum to the cell membrane really depends on palmitylation, because if you don't have the palmitylated form, there is a decrease in the association with the plasma membrane associated NOX5. And this became especially exciting to us because we had published in 2020 that NOX5 very closely co-localizes with cholesterol-rich microdomains or caveoli or lipid rafts within the plasma membrane. And we did this by a number of investigations looking at quantification as well as qualitative studies using imaging. But we can see that if we fractionate our cells and we look at NOX5 in those fractions that are lipid rich, there's a very close association between NOX5 and the membrane fractions that are rich in caveolin 1 and flotilin. And putting a huge amount of work together, we went on to define that NOX5, once it is translocated from the endoplasmic reticulum to the plasma membrane, associates with caveoli and lipid rafts, where it is in very close association with many signaling molecules that are highly regulated by redox changes. One of which is CSARC. And CSARC is a caveolin-associated signaling molecule, highly sensitive to changes in redox status and implicated in many vascular smooth muscle cell processes. And I'll show you here just some examples where we're looking at actin polymerization, cell proliferation, and migration in cells from normotensive and hypertensive individuals that have been exposed to angiotensin in the absence and the presence of a CSARC inhibitor called PP2. We can see that many of the processes that are amplified and NOX5 dependent in hypertension are indeed attenuated when we reduce the activation of CSARC, as we can see here with PP2. For cytoskeletal organization, migration, and also proliferation, as well as calcium transients. So indeed, we're beginning then to put together a picture whereby NOX5, through its production of reactive oxygen species, is regulating many signaling processes that regulate vascular smooth muscle cell function. But let's get back to that vascular phenotype that I mentioned when we started this presentation in the context of hypertension. We know that vascular smooth muscle cells that contribute to this vascular remodeling are extremely dynamic and very plastic. And they have this amazing ability to undergo this process called dedifferentiation or phenotypic switching. We know that in pathological conditions, a contractile vascular smooth muscle cell can dedifferentiate into a synthetic, proliferative, pro-inflammatory phenotype. And it's also been suggested that reactive oxygen species do play a role in this process. So we went on then, again, in the notion of human vascular pathology, to look at some of the transcription factors that are important in this dedifferentiation process by targeting against some of those signaling pathways that I've just shared with you. We looked at the role of ER stress. We looked at the role, importantly, of NOX5 in this process of dedifferentiation. And what we can see is that some of the transcription factors, such as KLP4, which is very important in the phenotypic switching of vascular smooth muscle cells, a transcription factor that is increased in hypertension is indeed attenuated when we reduce NOX5, as well as if we look at inhibitors of ER stress. And similarly, too, if we look at the proliferative phenotype, looking at PCNA of vascular smooth muscle cells, this again is attenuated if we inhibit NOX5 with an siRNA, or if we look at ER stress inhibitors. So putting together again now the functional association between ER stress, NOX5, reactive oxygen species, and vascular smooth muscle cell function. So I've shared with you some molecular studies that we've used to really identify what is happening in the context of vascular smooth muscle cell biology. But this doesn't really tell us what is happening in the intact whole system. And as I said, because NOX5 is deficient in mice and in rats, it's very difficult to study NOX5 in the lab. But nevertheless, when I was still here in the Kidney Research Center working with Dr. Kennedy, we started to generate some NOX5 transgenic mice that expressed NOX5 in a cell-specific manner. We created mice that expressed NOX5 in the kidney, and these mice were found to be hypertensive. We've expressed NOX5 in the endothelium, in vascular smooth muscle cells, as well as in cardiomyocytes. And what happened is when we expressed human NOX5 in vascular smooth muscle cells in mice, we found that these mice did have a blood pressure signature, and that when we looked at 24-hour ambulatory blood pressure monitoring, and we found that their blood pressure was indeed higher than that compared to mice who did not have the NOX5 in their vascular smooth muscle cells. This was associated with oxidative stress, as we can see an increase in superoxide production and a pro-inflammatory phenotype. Moreover, when we looked at the vascular remodeling of these NOX5 containing mice, compared to those mice, the wild type mice, again, we see a huge amount of collagen deposition, an increase in the vascular media, and these vessels also express endothelial dysfunction. And when we started to look again at the collagen content of these vessels, a huge increase in the NOX5 expressing mice, as well as an increase in the markers of vascular smooth muscle cell dedifferentiation, as we can see here by the alpha smooth muscle actin and microde RNA expression. And what we actually think is happening in the in vivo system is that you're getting dedifferentiation to a myofibroblast type cell that is now expressing the NOX5, producing reactive oxygen species, and contributing to this phenotype. What about the heart? Remember, NOX5 is activated by calcium. Calcium is so important for vasoconstriction and also for cardiac contractility. So we did some collaborative work with colleagues in China, actually, and they created this mice where NOX5 was expressed in the cardiomyocytes. And we found that when these mice were exposed to cardiac overload, they had a profound increase in the remodeling, as well as a profound increase in cardiac dysfunction and hypertrophy, mimicking what we had seen in the vasculature. So it's very nice to see that this is happening when we take the human NOX5, put it into experimental models, but how does this really impact in terms of human pathology? Well, these were very nice studies that were done by our colleagues, Harold Schmidt, and he started to look at NOX5 and its signaling, particularly in the endothelium in human hypertension. And when they did in silico modeling, they found that NOX5 of the NOX family was the most important NADPH oxidase implicated in hypertension as well as in diabetic nephropathy, similar to what we had previously discussed. They also went on to look at endothelial microparticles containing NOX5 and found that these endothelial microparticles that expressed NOX5 were increased, especially in hypertensive patients with underlying kidney disease. More interestingly, we were very excited when a paper came out in the literature in 2017, looking at meta-analysis GWAS study of almost 500,000 individuals, and there was a very new association between this blood pressure associated loci that identified NOX4 and NOX5 as an associated new gene with respect to blood pressure, further confirming to us that indeed this may be an important process involved in the pathology associated with hypertension. So I've shared with you now some of the work that we have done related to putting NOX5 on the map of the vascular biology implicated in hypertension. We know that NOX5 produces reactive oxygen species, and that when this is uncontrolled in pathological conditions, we have oxidative stress. But at the end of the day, how does oxidative stress or an increase in the bioavailability of reactive oxygen species in cells actually have an impact on the downstream signaling molecules, some of which I've shared with you? We know that transcription factors, that ion channels, kinases, protein tyrosine phosphatases, genes are very, very tightly regulated by the redox state of cells. And we believe that this is due to the oxidative post-translational modification that these proteins undergo when you get an increase in oxidative stress. And I just want to highlight to you that post-translational oxidative modification is actually a very complex process, but there are two components related to this. There's a reversible oxidation and an irreversible oxidation. And we believe that in pathological conditions where you start getting irreversible fibrosis, irreversible cardiac vascular remodeling, the cells and the signaling, the molecules have undergone the irreversible oxidation. It's sort of the last point of cell dysfunction. So when we started to probe then oxidation of the proteins in hypertension, we published a few years ago that indeed there is an increase in the oxidation of proteins, and this is looking at a PTP in cells from hypertensive patients. Again, an increase in the irreversible protein tyrosine phosphatase oxidation. And more recently now, we have gone on to actually identify the proteome that defines those abnormalities associated with vascular smooth muscle cell dysfunction. And we've started to put together through gene ontology those proteins that are differentially expressed between normotensive and hypertensive individuals. And again, not surprisingly, many of the systems that are panning out really relate to inflammation, connective tissue, and remodeling. But very excitingly, we've taken now this vascular proteome and we've started to look at the oxidative processes that are particularly linked to those changes that occur in the alterations of the proteome in hypertension. And our current work is now focusing on identifying the oxidative proteome in hypertension. And we now have identified a number of proteins that are oxidized, that are reduced, and that are irreversibly oxidized compared to reversibly oxidized, again, in human hypertension. We think this is important because at the end of the day, as I say, it is these proteins in their post-translational form that will ultimately impact cardiovascular function. So,
It's getting quite excited because a number of those pathways, we do a lot of biomarker. they show up and like,
oh,
yeah, I see the interferon, stimulation.
yeah, yeah.
Of course, you know, we do a lot more IGF.
Exactly, yes. Yeah. Yes.
Maybe we can start from 39th slide.
Yeah.
Okay.
Oh, it's loading.
Yeah.
It's too many proteins. Can we connect my computer maybe?
No. So it's using connect your USB. So right now I already
copied it.
Yeah. Computer, but these slides cannot be shown. Yeah, see, it's blanked.
So what should we do?
Just try to reopen.
Sorry about this. I've never had that before.
Yeah, our technology limitations.
Oh,
yeah. Up to the latest.
This has happened to us in teaching before.
Oh, it's happened before.
If you slide it on the screen.
I don't know if we
You have to connect the Zoom for this. Let's try to copy your presentation again using this USB stick.
I've got a connection. Good seeing you
Yeah.
too, and congratulations on
Yes, yes.
exciting.
Yeah, I have to run because I'm on the.
Sure.
But I probably am gonna want to pick your brain soon. Yeah, and maybe I'll give you a shout separately.
Absolutely. Pleasure. Pleasure. And all the best.
I do have a question though. I mean, it doesn't have to be for now, but whether the vascular changes are actually the most things like targeting and non-spot, and that
That we think that presenting translation of after. We think that indeed some of the mechanisms we're talking about, because it reduces interference or reduces non-spot activity. That's exactly what we tested about. Is it still not there?
Still not loading.
Not loading.
It's already copied there.
Do we check to see if that it's still on here?
It's here.
Another stick.
I have to reboot the whole computer. Yeah, it's completely frozen. So computer.
Sure.
To complete the whole presentation.
Sure.
And after that, I fix this.
Okay, sorry about all of that, but anyway, there we go. So the paradigm that we're working on, as I said very briefly, is here. We feel that The NOx5 plays a really important role in terms of its crosstalk between reactive oxygen species calcium signaling, the oxidation that takes place in signaling, and ultimately altered vascular function, leading to small vessel disease. However, as we know, it's becoming much more evident, and we've realized this over the past few years, that hypertension is not only associated with cardiac disease, heart failure, ischemic heart disease, chronic kidney disease, and stroke, but there's now a real, there's very clear evidence that as blood pressure increases over the course of one's life span, this is associated with an increase in the risk of developing vascular dementia. And we know that the burden of vascular dementia in terms of personal, societal, public health is huge. And we know, as I said, and now the evidence is really growing, especially in the midlife, that as blood pressure increases, so too is an associated increase in the risk of vascular dementia. And this is very nicely seen in this study, and that was published in Hypertension a few years ago. So when I was again in Glasgow, I had the fantastic opportunity to work with our neurology colleagues who had a specialized clinic looking at patients with small vessel disease of the brain, and we started to, let's see, we need action, it seems here, what is happening? A wonderful opportunity to work with our colleagues in neurology, and they were looking at patients who had subcortical infarcts and leukoencephalopathy. This is a congenital autosomal dominant condition, a condition called CADASIL. And for those of you who don't know what CADASIL is, it's a small vessel disease of the brain. It is characterized by premature vascular dementia. It is the most common cause for monogenetic forms of stroke. The prevalence is not that low in populations. And the thing is, we know the cause of CADASIL, and the cause is a mutation in the NOTCH3 gene. And the sad thing about it is that patients die very young, about 10 to 20 years after the onset of the symptoms. And if we look at the vasculature, the small vessels in the brain, we see very similar changes to what we see in patients with hypertension. They undergo this very significant vascular remodeling. There is an associated alteration in endothelial dysfunction, and this is associated with vascular smooth muscle cell disorganization. The very typical in CADASIL is the deposition of this abnormal protein called granular osmophilic material deposits, which is actually aggregates of the NOTCH3 protein that have undergone abnormal processing. Now, as I said, NOTCH3 mutations are responsible for CADASIL. NOTCH3 forms part of a family of 4 Notch isoforms. These are very complex receptors that have very common EGF-like binding domains in their extracellular domain. But it is the mutations in NOTCH3 that are responsible for CADASIL. Now, the thing that is especially relevant with respect to NOTCH3 is that NOTCH3 is exclusively expressed in vascular smooth muscle cells. NOTCH3 is not found in other types of cells except perhaps in pericytes. And this for us became exceptionally interesting because it's a vascular smooth muscle cell specific receptor. It plays a role in vascular small vessel disease. And so we were very intrigued to try and start dissecting out some of those signaling pathways that we had identified in patients with hypertension. And we asked a few very simple questions. And that was if CADASIL is associated with a NOTCH3 mutation, this mutation is ubiquitous. It's not only in the small vessels of the brain, therefore may there be peripheral small vessel disease as well. And we started to interrogate that. And as I said, we started to gain some insights into the molecular mechanisms underlying NOTCH3 mediated vascular injury. And most importantly, we wanted to identify putatable druggable targets because there are no treatments for CADASIL, this this terrible disease that affects families, some of the vascular function and endothelial dysfunction through modulation of the oxidative stress. We published that in experimental models, but actually we have opportunities now in our patients who undergo rehabilitation to look at some of this as well. Absolutely, yeah.
Great, yeah. And certainly we have excellent exercise programs here.
Yeah,
that can collaborate on that. And maybe last question from Katie Rayner. So wonderful presentation. Are there less severe forms or dysfunction of Notch 3 that being identified in microvascular dysfunction in the brain?
Yeah, so the carousel is really interesting. There are a whole host of different, I think there are about over 20 mutations that are identified with the Notch 3 mutation. And I think some of the mutations are more severe than others. The other thing that's very interesting is that the patients that we studied had a gain of function of Notch 3. So the abnormal Notch 3 was associated with an increased activity of Notch 3. There are some patients where the mutation is actually an inhibition of Notch 3. So there is variability, absolutely.
Right. Okay. So there are nature's models, from that point of view. Yeah. Fantastic. So, I know in view of the time, and so really appreciate Dr. Rhian Touyz to really have the wonderful opening lecture for the this academic season, and apologies, for the technical challenges. But I think it really illustrates the wonderful science, of the ability for us to actually ask clinical questions and take that and dissect out in, specific models at the cellular, even the proteomic models, and take it back to the patient. This is really translational cycle is really, sort of fantastic model of how we can unpack solutions, for clinical challenges and address the cardiovascular problems and the brain and the heart as well in terms of the programs that we have in collaboration. Looking forward, to more discussions and opportunities. And thank everybody for joining us both here as well as online. And for some of the folks, who may want to have a little chat, with Rhian, we have a room set up for that. So thanks so much again, and thanks very much, everybody.